Good morning. How are you?
Good. How about you?
Pretty good. Let's see. I want to do a screen share. Okay. And I'm going to move this to the bottom because it's always in my way at the top. You got it? Awesome. Okay, you got to fill out part of it.
I was going to do it after.
We'll just at least put your name on it. Okay, let's see. Go to drive. Get a front row seat today.
I don't want to steal anybody's seat.
I think this is Friday. There are no you can steal anyone's seat.
No assigned seat on Friday.
That's right. Okay. Of course, got to do two-step verification. Yes, it's on. Okay. $10, $16. Good luck. Oh, thank you. All right, sounds good. Yeah. Okay, so here we go. I want so many different things. All right, let's see. And the same thing. Notebook file, so it should open normally. Fingers crossed. And I need to open Clicker. It's like how many things to open. Good morning. So nice to see you. I love seeing people on Friday. Makes my Friday better.
It's almost not as bad to come. It's Saturday.
Because it's Friday. It's not a
This is
big
the only
deal.
class I have today, so I have all this up.
It's awesome.
Yeah.
You got a bunch of classes today?
Just two.
Just two. It's not bad. I got lab after this, so it's a long day for me.
Yeah, that's not what it is.
I kind of like my Friday lab. I think it's fun. We're all pretty focused, right? In and out. Here we go. And I need attendance.
Do we get on Zoom in class?
If you want to, you can, but you don't have to. It's not a requirement. Okay, I want my attendance. I think I left it in here. No, that's not right. This one. Here we go. I know I had it here somewhere. Okay. Oh, and I need to turn my chat on. I'm so sorry. I see y'all are chatting with me and I didn't. What happened to your face? Oh, yeah. I just walked over with my mask on. All right, let's see. I'm going to minimize this and I will turn on. Nope. Oh, there it is. I didn't even see it. Okay, so you can do your attendance now. All right, my list of things to talk about. List here somewhere. Oh, please tell me I didn't leave it. Here we go. Here we go. Okay. I want this one. Okay, that's good. But I do want to pick what slide because we're We're pretty far down. What number is this? No. 8.3. Oh no, I went too far. Here we go. Okay. Y'all are so patient. I'm almost ready. I'm gonna need my chat back. Oh. My chat. Okay. Hey. Okay. So before we start our regular stuff, I talked to Dr. Smith today. We were discussing the lab final. And so the lab final is going to be online. So you do not have to have a particular day. We're going to figure out exactly when we can do it. We're not sure which days it will be, but it'll be online. So you don't have to be like on campus and that kind of thing.
So online multiple choice.
It will be online open lab notebook.
Oh.
Yes, yes. So anything you have written in your lab notebook, that's why we kept telling y'all from the beginning, write as much as you can in your lab notebook, right? Yes, our exam is going to be on Wednesday. Our exam is still going to be on Wednesday, yes. Okay, so our plan, right, our general plan, let me see if I can just extend the page. I'm going to go back one page and extend that page. That way I'm not messing with this. Why can't I do that? I want to do, okay, anyway, all right, so here we go. So we have today is Friday, come on. Today is Friday, so we're going to do 8.3 today, right? Then Monday, I made the video for Monday. It is 10 minutes long on 8.4. In class, we'll do 8.5. This will be class. But I'm telling you that 8.4 and 8.5 are short. They're very short. And I'm not going to expect the detail that I expect from 8.2 and 8.3. So the question at the end of the last class was what cascades or what signaling sequences are you really going to have to know, right? So from 8.1, you had your phosphoanesthetize, PHOS, anestetide cascade, right? And that's going to be important for other systems, right? You see it in the GPCR, your G protein signaling, when you go through PKA, right? And then also in, you have to know your G protein cycle. So that's from 8.2. Then 8.3, it's your receptor tyrosine kinases, which is what we're going to talk about today. And there are two of them. You have to know EGFR and insulin. Those are the ones that you need detail on. The others, when we talk about TNF alpha, which is the tumor necrosis factor, you don't need to know a lot of detail. That's going to be really general. And the last one, which is your nuclear receptors, that's going to be really general. So as of today, you'll have all the detailed ones. So you'll have this weekend to study the detailed ones. Okay? Good? All right. So my hope is that Monday, it won't take us very long at all to do 8.5, and that we'll have some time to work on a few clicker questions with those more detailed pathways. And so what I did, I don't know that we'll cover it all, but what I did was I made, did I post it? I think I posted it. I wanted to post it. Let me see if I did. 8.0, no, it's 8.3. I'm gonna post it when I leave here. I'm gonna post it. It'll go right here. It's the section, it's like section 3 discussion questions. I must have put it in Google Drive and not put the link. So I will go ahead and put the link when I walk out. And that's what I want y'all to kind of look over so that we can answer some of those when you come back. They're like the long clicker questions. They're multiple choice. I'll show you. I will. So much. Here we go. I'll show you what it looks like because I have it. Files that are on Moodle. Here we go. Chapter 8. I'm going to do it right now. Copy link. Get link. I'm going to put it right now so that you're not waiting on me. Okay. Come on. Come on. It's not going to let me. Hello. All right, I will post it when we leave, but this is what it looks like. Right? These are the ones, this is similar to what we did with 8.1, right? And it just has some questions in it for you to kind of work through, you know, what we normally do in class. Okay, so on to the material for today. Any questions? And don't forget your clicker, not your clicker questions, your self-check questions are for points. Remember that? So you actually have to do them. But you get 3 tries and it's not timed. Tuesday night at 9 o'clock. So the night before the exam. Okay? What? No, this has been, we've had self-check questions like all year, but now I'm making them 10% of the grade. Yeah, that part, the 10% of your grade, that part's new, right? Okay, all right. So let's talk about receptor tyrosine kinase. So this is our second big, will this recording, yes. I'm gonna post all my Zooms. I always post all my Zooms. I'm gonna post this Zoom just like always. Okay. It's so big, but okay. I just can't see it all. All right, so receptor tyrosine kinases, Rtks. So this is your second major class of signaling proteins, right? So we're again going to transmit an extracellular signal through a ligand, right? The ligand binds, but in this case, we're going to have an intrinsic kinase activity in the receptor. So the receptor itself has kinase function. So the receptor has the ligand binding on the outside, and then it has a transmembrane domain, and then the cytoplasmic side is where we have the kinase. So in order to be active, a receptor tyrosine kinase has to become a dimer. It has to dimerize. Once that dimerization occurs, then you have all kinds of phosphorylation events that occur. This is why we call them receptor tyrosine kinases. What does a kinase do? Phosphorylates, right? Phosphorylates. Yes. So that's the whole, it's going to be all kinds of phosphorylation today, like phosphorylation. Okay. So once we have our receptor that binds our ligand, right? What are we trying to target, right? We have this active receptor that has a kinase activity inside of it, and it wants to phosphorylate things. What's it going to phosphorylate? It's going to phosphorylate adapter proteins. So an adapter protein is going to bridge the gap between our receptor and initiating downstream signaling events. That's what an adapter is, or they call it a molecular bridge. You know, we have to have another fancy name. And then a lot of times those adapter proteins are kinases as well. Like I said, lots of phosphorylation events, right? So what are some examples of receptor tyrosine kinase molecules? Well, the epidermal growth factor receptor, CEPT, receptor, or the insulin receptor, right? So what do you think the epidermal growth factor receptor's ligand is? The epidermal growth factor. What do you think the insulin receptor's ligand is? Insulin, woohoo, right? We finally named something well. We're gonna get to some things that they named very poorly today. I'm so sorry, but they did. All right, so how does this work? Well, we have a growth factor, right? And a growth factor is like a little protein that's secreted, right? It's paracrine and autocrine. Remember that? Right? So paracrine is close, far away, close,
But different.
but a different cell type, right? Autocrine is the same cell, right? Okay. And so a lot of times these growth factors are present during when you want to initiate cell division, right? You want to secrete something that's going to say, okay, you can divide right now. The environmental conditions are great for cell division, because eventually our downstream signaling pathways are going to promote that activity. So our growth factor binds to our inactive monomer. When that happens, we need to get a second growth factor to bind in a second receptor. Those receptors then dimerize. When those receptors dimerize, it ends up phosphorylating both of the original receptors. Once those are both phosphorylated, now we can bind some adapter proteins, and then we can phosphorylate some proteins, and we can activate our downstream signaling. So I want you to have kind of a brief overview of what we're going to do before we go into the big details, right? Okay, so, all right, so how do we do this? Well, EGF is a growth factor. So it's a growth factor found in our serum. And serum, right inside of your bloodstream, serum. So it binds to EGFR. It's going to stimulate the receptor, and the receptor is going to dimerize. So did I say that epidermal? Did I define it? Epidermal? No. Epidermal growth factor. That's where the E comes from. This works basically for any growth factors. And we have kind of a two-step model of that binding. So the ligand you have to have, and I don't like the way they did this. It's really, you need two ligands to bind to two EGFR receptors, right?
It's the same process for any growth factor.
Yep, pretty much. There are little variations, but not enough that you need to know them. All right, so you have two ligands that are going to bind two EGFR receptors, right? They're going to induce that dimerization. When the dimerization occurs, you're going to actually have a conformational change in the cytoplasmic tail, and one will get really close to the other. And so EGFR1, so the first, they kind of arbitrarily say which one it is going to phosphorylate EGFR2. So, and it's going to happen on tyrosine residues. What classification of receptors do we have? Do you know? Receptor tyrosine kinases. This is why. This is why we call them receptor tyrosine kinases, because they phosphorylate the tyrosine residues on the receptor. Okay, so then phosphorylation on EGFR2 induces a conformational change, right? We had binding that induced a conformational change. So then we had receptor 1 phosphorylating receptor 2. Now receptor 2 is phosphorylated, so we have a conformational change. And that conformational change allows EGFR2 to phosphorylate EGFR1. So now we have both receptors inside of the dimer being phosphorylated. So I'll show you what that looks like, right? Here's one, and it's not bound, and here's another, it's not bound. They match, right? Here comes our growth factor. Growth factor binds. Then one of the receptors is going to basically get really close to the other one and get phosphorylated. So we have our first one becoming phosphorylated. That's why we call it EGFR1. And once that becomes phosphorylated, now that kinase activity can become active, and we can phosphorylate EGFR. Well, EGFR2 can phosphorylate 1, right? Because 2 was the one that became phosphorylated first. I know it's too many numbers in first and seconds.
So EGFR1, will it ever be phosphorylated before the second one?
It's arbitrary which one is first and which one is second. But when we say EGFR1 is the one that does the phosphorylation event first. So it's the one that is conducting the first phosphorylation. EGFR2 is the one that conducts the second phosphorylation event. Okay. So our head domains look pretty much the same, right? In here, the conformational change that occurs because of binding occurs in those cytoplasmic tails, which is what allows that phosphorylation event to begin, right? Okay, and then it's all about every time we have a change and an event can happen, it's because of an induced conformational change. So you have to remember that all of this is an allosteric regulation. So all of this is allosteric LAT regulation, right? One thing binds, causes a conformational change, something can happen. That's allosteric regulation. So our dimerization regulates tyrosine phosphorylation. So you have dimerization controls our first phosphorylation event. Yeah, and then the first phosphorylation event controls the second phosphorylation event. And so on and so on, and we're going to phosphorylate some more stuff today. You didn't know how much phosphorylation could happen, right? Okay, so now we have these phosphotyrosine residues. This is the symbol for a phosphorylated tyrosine, right? So here and here we have phosphorylated tyrosines. So these residues are binding sites for our adapter proteins. So here's an example of an adapter protein. An adapter protein is going to bridge the gap between our receptor and the next signaling molecule. So our first signaling molecule is called GRB2. It's growth factor receptor bound 2. Growth factor receptor bound 2. That's the name of the protein. Now, in order for any protein to bind to phosphorylated things, they have to have a common domain. There has to be a part of the protein that recognizes a phosphorylated residue. So right here, this little portion of the protein has an SH2 domain. An SH2 domain, by definition, will recognize a phosphorylated tyrosine, and it's going to recognize some other residue a certain number away from that phosphorylated tyrosine. So it could be a phosphorylated tyrosine and 2 residues away an asparagine. Or it could be a phosphorylated tyrosine and 3 residues away, it's something else, right? And so this allows for really good specificity in terms of, is the receptor active and which receptor you are going to bind to. So it gives you those 2 pieces of information. It's not just any old phosphorylated tyrosine, because a lot of that's floating around in the cell. All right, so now our next connector. So once we have that GRB2 that's bound to our phosphorylated tyrosine on the receptor, then that is going to recruit our next protein in the sequence, and our next protein is SOS. This one is named absolutely ridiculously. It's the son of sevenless. The son of sevenless. Why? I don't know.
There's
Maybe, I don't know. Some people just like to name things really crazily. They do, they do. The son of sevenless. Woo. All right, so once the son of sevenless is recruited, right? This is a GEF protein. What does GEF stand for? Do we need to know the whole names? You can just know the abbreviations. The adapter protein bridges the gap. No, GRB2 is the adapter protein. GRB2 is the adapter protein that bridges the gap between the activated receptor and all the downstream signaling events. Does that make sense, Taylor? Yes, okay. So once we have our GRB2 adapter protein attached to our phosphorylated tyrosine, we're gonna recruit son of sevenless. Son of sevenless is really cool because it is a guanine exchange factor protein, right? So what do you think a guanine exchange factor protein does? Exchanges.
The last section wasn't it like it changes GTP for GTP?
Yeah, look at this. And so our next binding protein is RAS. RAS has a site where GDP can dock. So normally in the inactive form, RAS has GTP. When it is associated with a guanine exchange factor, what do you think it's going to do? It's going to exchange, right? And so now we're going to have RAS with GTP. So RAS is an example of a G protein. Remember from last section, G proteins, right? So it's in the same class of proteins. So this is something you've already been exposed to. All right, so that's great. So now this is active RAS. This is active. Active. It can initiate all kinds of downstream craziness to happen. Now, if you left it like this, RAS would signal forever. It would be stuck in the on position. I'm telling you, that's cancer. So we have to have in the cells a way to turn it off. So our way to turn it off is we have a gap. All right, so a gap protein is a GTPase activating protein. That's what the GAP stands for. GTPase activating protein. So when RAS GAP binds to RAS, it facilitates the exchange back to the inactive form. So this is inactive. So that RAS can be turned on, initiate signaling, turn it off again. You want it to happen quickly because once you initiate the downstream events, they're going to go. You don't want them to continue to go. Okay? All right, so our signal, so it results in a short-lived signal, which is super important. Okay, so now what happens in all this downstream signaling pathway, right? That's the next thing we need to figure out. Like, what do I click? All right, so this is kind of just defining the things that we already said. The RAS protein is a member of our G protein signaling molecule. It's like G alpha. It's a type of G alpha. And we saw that it was anchored to the cytoplasm, right? So we have it anchored into the membrane. And it's covalently attached to, that's the lipid that anchors it, right? It's activated by, what's GEF stand for? Guanine exchange factors. It contains an intrinsic GTPase activity that are regulated by guanine, not guanine, oh my gosh, by GTPase activating activating proteins. Right? There's a lot of abbreviations, I know. But every section in this book has like a table of all the abbreviations, the definition of the abbreviation, and the function of that protein. So use that. Absolutely use that.
So it's kind of like the same as the second part with the G protein is the same as the last one.
It's very
Except
similar.
the beginning is different.
The beginning. Yep. 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 It all kind of builds on itself. All right. So now we got to kind of figure out how does RAS, once RAS is activated, right? It's in its active form. It's got GTP in it. So how does this initiate downstream effects? So we're gonna part of it gets glossed over, but that's okay because I think it goes into plenty enough detail. So we're gonna assume we know how to get to activated RAS. We're in the RAS GTP form. So we're gonna recruit some other proteins that are gonna initiate a phosphorylation cascade. So we're gonna recruit, I know. I know. It's a lot of, I told you a lot of phosphorylation is gonna happen today, a lot of phosphorylation. The whole point of these phosphorylations are to initiate what are called the MAP kinases. And the MAP kinases are kind of a big group of proteins that get active and initiate a big series of events. It's not just like one activity gets activated, it's a big series. And so, so if we look at RAS, right? RAS, once it is in its GTP form, it's gonna recruit RAF, it's going to be phosphorylated by SARC, it's gonna initiate this downstream effects. And when we have a mutation in RAS, right, this is where we can lead to cancer. When we can activate RAS and leave it active, this is a huge problem. Because if you have a mutant RAS protein, this is going to be a constant activation, activation of our kinase cascade. If our kinase cascade is going on and on and on and on and on, then you can lead to mutations, you can lead to cancer and all that kind of stuff. So have y'all heard of something that's a mitogen? Have you heard that term before? No? Sometimes you'll hear that as defining things that can be permanently turned on and lead to cancer, because mitosis is cell division. So a mitogen is something that continuously activates cell division. So I don't know if maybe y'all had heard that before. Okay, so I kind of want, before we turn and talk about insulin signaling, I want to go back and I want to see if we can kind of write out a pathway. And y'all can tell me what happens in order to get to our signaling cascade, okay? So if we're gonna initiate this, I have a growth factor ligand, right? What's it gonna bind to? It's gonna bind to the EGFR1, and what else does it have to do? Bind to EGFR2. That binding induces what change? Anybody at home can join in. Dimerization, that is correct. When these two dimerize, what's the result? 2 is phosphorylated by 1, or 1 phosphorylates 2, however you want to say that. So let's say EGFR1 phosphorylates EGF. Why do I keep 2? Right? Okay, now what happens?
two phosphorylates. One.
So then when I say, I always try to say that it's phosphorylated. So phospho, EGFR, 2, right? Phosphorylates, EGFR1. So now we have both of them being phosphorylated. Those phosphorylation events do what? What? Well, they're going to recruit something. It does cause a so each one of these things, dimerization causes a conformational change. EGFR1 phosphorylation of EGFR2 causes a conformational change. Then the next step causes a conformational change. They all cause conformational change. They're all allosterically regulated. They let, Adele's right, they let adapter proteins bind. So they recruit and bind adapter proteins. Okay, once we've recruited our adapter proteins, what is our adapter protein? GRUB, right? GRB2. Our GRB2 is going to recruit and bind what? SOS, son of sevenless. So crazy. What is son of sevenless?
GEF.
Yeah, a GEF, which is a guanine exchange factor. Okay, so that's great. It's an exchange factor. What does it exchange? On what molecule? On RAS, SOS can induce, what you got?
The domain SH2, what does it do?
The SH2, okay, here on G, the question is the SH2 domain on GRB2, what does it do? SH2 domain recognizes the phosphorylated tyrosine residues on, gotta move this, on the EGFR. And then it has SH3, it actually also has SH3 that are gonna bind to SOS, right? That's why it's an adapter protein, because it can bind to both the receptor and to SOS. So it's an adapter. Okay, so we have our adapter, right? Our adapter binds and recruits son of sevenless, which is a GIF, which is a guanine exchange factor. The guanine exchange factor works on RAS. What is RAS? A G protein. Our G protein here, right, can induce, I'm sorry. So on RAS, SOS is going to induce a change of what? GDP for GTP. So now we have active RAS. Active RAS has GTP. Now, two things are going to happen. Whoa. We're going to initiate downstream signaling at the same time that we're going to basically turn it off. So we're going to activate our downstream signaling cascade, right, through all kinds of phosphorylation events. So RAS phosphorylates, RAF phosphorylates, MEK phosphorylates. Like, you could go on and list a bunch of proteins that get phosphorylated, but I really just want you to know that it activates a downstream signaling cascade, okay? Now, once we activate that downstream signaling cascade, if we don't turn it off, we got a big problem, right? So what are we going to do? We need to inactivate it. We need to stop it. So what are we going to stop it with? We're going to stop it with a GAP. So a GAP, what's a GAP? Let me move this over. A GTPase activating protein. Okay, so our GTPase activating protein, right, is going to make RAS GTP turn into what? RAS GDP. RAS GTP is our active form. GDP is our inactive form.
It doesn't get turned off, mutations get there, or
Well, if it doesn't get turned off, usually the problem is you've had a mutation, and the mutation has disrupted this stop from happening. So maybe it's a mutation in RAS, maybe it's a mutation in the gap. It doesn't matter, but you would have a mutation in one of those two places. And so if you mutate those two things, the stop never occurs. And so basically you get this cell signal that says divide, 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 divide. It never stops, just never stops. And that's a cancer cell. So a lot of the cancers that you're going to find will happen with mutations in proteins that control the cell cycle, right? And that's what you're controlling here. You're eventually going to control the cell cycle. You're eventually going to get to, you know, you do through all these phosphorylation events, and you eventually get to regulating transcription factors. And those transcription factors are going to control the cell cycle. You're going to say, turn on, turn off this gene. Genes that are important for cell cycling and all that kind of stuff. So are y'all familiar with oncogenes? Have y'all heard that before? Anybody on the chat heard of oncogenes before? Yes, yes, ooh, yes. And tumor suppressor genes? Yeah, okay. So if you have a oncogene, right, that's gonna be a dominant mutation where you're gonna have a gain of function. And that's exactly what we've been talking about right now, right? You're gaining the function of inducing transcription of all of this stuff. So that's going to lead to a diseased phenotype, right? So RAS is actually the protein that is mutated in the most common forms of cancer. I feel like this is probably the interesting part of this, right? How it applies to cancer. And so when you mutate RAS, things keep going. You have a gain of function, right? So RAS mutations are a gain of function. So RAS mutations, gain of function. Let me move this over. Gain of function. Right?
would that mutate the RAS be considered oncogene?
Would be considered the oncogene, O-N-C-O, the oncogene. Because this is the one that got mutated in order to cause cancer, right? If you're a physician and you study people who have cancer, what do they call you? An oncologist. So that's why we call it an oncogene. It's a gene that, if mutated, will induce, to become active, will induce cancer formation. So all of the genes that regulate cell cycle control are called mitogens. But oncogenes are ones that, if you mutate them, will turn on cell division. It's like you're in the car and you jammed your foot on the gas pedal and you can't take it off, right? That's a cell in cell cycle just going and going and going. Okay, and then the other one is the recessive mutation, where it's like if you had a mutation in this gap. If you mutated this gap, let's say, you know, you have two genes, one from mom, one from dad, right? If you mutate one of the gaps, you have a backup. and that can still stop this signaling pathway. But if you mutate them both and you take them away, it's a loss of function. And so you end up also with cancer because you can't stop, but for a different reason, right? That's the difference between a gain of function and a loss of function mutation. They're pretty cool. I could study that for, I don't know. There's so much in biology, biochemistry, whatever, that I could just get lost in. All right, so let's move on. Let's talk about another receptor tyrosine kinase. So insulin signaling is another receptor tyrosine kinase example, right? And this one's going to be really, really important because we're going to talk about the regulation of insulin signaling in like a chapter. So this one's really important. So our ligand, thankfully, is called insulin, and the receptor is called the insulin receptor. Woohoo! And it, guess what it induces? A conformational change, right? So this is another example of allosteric control. So we're allosterically changing the conformation. So when we look at a diagram of our receptor molecule, right? We have two alpha subunits. So here's one of the alpha subunits. Here's another alpha subunit. And then we have our beta subunits down here at the bottom. And so I don't really like so much their drawing here, but that's okay. It originally comes from, you know how we've talked about proteins are peptides that are held together by disulfide bonds. Remember that? So this is a good example of that. You had one really long peptide that actually got cleaved into 4 pieces and then joined together again with disulfide bonds to hold it in the right configuration to act as a receptor. So this is a good example of what we talked about before. Okay. All right, so what happens in our insulin receptor signaling? Oh no. When I convert it to this form, I lose my link. That's so sad. All right, I will post this. That's so sad. I can't believe I lost the link. Okay, I will post this video. That's so sad. But this is, I like this diagram a little bit better. This is a really great animation of, this is the extracellular surface, right? You have your transmembrane domain, and then you have your beta subunits that are sticking out. And these little, you see the little yellow things on there? Those are phosphorylation sites. And so you'll see how it interacts. Oh, that's so sad. So sad. Okay, so we'll work off of this diagram here, right? We have our alpha subunits, and our alpha subunits are what actually bind insulin. Insulin binding is a little bit different. Remember, before when we had the growth factor, we had to have 2 growth factors bind and induce dimerization. Well, in this case, everything's all kind of linked. And so when insulin binds to one of the alpha subunits, it actually inhibits binding to the other subunit. So you only need one insulin molecule in order to initiate this pathway, right? So your one insulin molecule binds to your alpha. It induces a conformational change, right? You're going to induce a conformational change in these beta subunits. So now we can affect the catalytic activity of the beta subunits. So now we're going to actually undergo autophosphorylation. So what's the difference between autophosphorylation and what we did before? Right, so beta 1 phosphorylates beta 1. Beta 2 phosphorylates beta 2. And that autophosphorylation event was caused by the allosteric control when insulin bound to alpha 1. Okay, so there are 2 different pathways that get activated. Look at this. We have one that's super familiar, right? I see RAS, I see son of 7 list, GRB2. There's just one different adapter protein here, and that's SHC. It's the sarcomology collagen protein. It's okay. but it's another adapter protein. That's what you just have to know. It's another adapter protein. And we can stimulate that same pathway, and look what event we have. MAP kinase signaling proteins get activated. We go through cell division, right? This is the same shared pathway that we did before. We just have a slightly different adapter protein. That's why adapter proteins are specific to their receptors. So they can initiate the same downstream effects, but based on different ligand binding. Okay, so now the second pathway that can happen, you got to think about what's insulin for. You just ate a meal, right? Something delicious. And your body started to break down your lunch. And you now have high blood sugar. This is good, right? Because that blood sugar is going to be the fuel for all of your cells. But it's sitting in your bloodstream. It's not in your cell. You need to get it into the cell. So insulin is the hormone that triggers the cell to say, ooh, there's glucose out there. Let me go get it. And it pulls it in. So eventually we want this pathway to stimulate glucose uptake. Now, a lot of times, and I'm guilty of it myself, I eat more than I need, right? Well, guess what I'm doing? I'm doing glycogen synthesis. Glycogen is a storage molecule. Y'all know that, right? So we can store glucose in the form of glycogen. So we do that a lot of times in our liver, right? So that, like, if you're in a fight or flight response and you need blood sugar right now, your body can initiate degradation of glycogen, and you can run from the bear that just entered the back of the classroom. Whatever it is. No bear actually entered the classroom, I promise. All right, so what happens in this pathway? In this pathway, right, we have our phosphorylation event that's going to recruit an adapter protein. Our adapter protein in this case is IRS. IRS, I know, but it's insulin receptor substrate. So that one actually makes sense, insulin receptor substrate. Our insulin receptor substrate gets recruited and phosphorylated, right? When we phosphorylate this adapter protein, we're going to recruit PI3K. PI3K is the phosphoinositase kinase, right? So it's going to convert PIP2 to PIP3. It's just going to add a phosphate group, right? That's what the 3 and the 2 are. They're just phosphates. Remember, we did something different with PIP2 before? We split it. Exactly, into DAG and IP3. So now we're just doing something different with the molecule. And we're going to initiate a different downstream event. Okay? So here's our IRS, right? Our IRS gets phosphorylated and activated. It's going to bind to our same PI3K. I don't know why. Oh, they did. Okay, so it's still PI3K. We're just making it look different because it's inactive. Now it's active, right? Our active PIP3 is going to take our PIP2 that only has 2 phosphate groups on it, and it's going to attach another phosphate to it. So now that we have PIP3, this is the same PIP3 that we talked about when, let me move this over, when we talked about PLC, our phospholipase C, remember that? Let me move this where I can still read, okay? Yes, good, good, good. All right, so now our PIP3 can be a substrate for another kinase, right? Aren't y'all so excited to hear about more kinases? Which can recruit what's called AKT. AKT is another one of those kinases, and the event is that we end up with glucose intake and we make glycogen, right? That's our whole point, so that we can initiate those downstream events. And so it's all about phosphorylation, but you need to have a stop, right? Just like we talked about before, you have to have a stop. Here's our stop. We have a new protein here called P10. P10 is what is going to halt our signaling cascade, right? and it's going to make it go in reverse. So here's our stop point. We always have to have some point where we can end the cycle. So our PI3K signaling will stop. Our PI3K here will stop. Wait, is IRS even taxing? Yes. Our PI3K will stop phosphorylating PIP2 in the presence of P10. So P10 will just end the cycle. Nothing is off limits for the IRS. It's true. Absolutely true. What you got? I see a question.
I'm confused. When you go from PI3K all the way to
PI3K, we're activating the kinase to what?
all the way to glucose uptake.
Mhm.
How does it know to stop? Like, does it give you the glycogens and then stop on its own, or does it still give it a signal?
So you will continue to make insulin as long as you have high blood sugar, right? So this is going to be a quick stop, just like before. So in order to continue to uptake glucose, you have to continue to get insulin signaling, because you want to stop taking up glucose when your blood sugar gets too low, right? So it's the same kind of quick stop, but for a different reason. But the same thing is true, yes. Okay, so the question was like, when does P10 stop the signaling? And it always does, because you want to only activate the signal when insulin receptor is bound to its ligand. So when your blood sugar drops, you'll stop making insulin, and so you'll stop activating the receptor, and all of this will end, right? Okay, I think that's it. Well, look, and I had 2 minutes to spare. So my suggestion to you is, you know how we did that? I did that blank sheet, and I said, okay, how does it go? What are the steps? Start doing that. Maybe do it one time with the book open, right? With your notes open. Do it one time. Then get a dry erase board, a blank sheet of paper, and practice. So this is the end of the signaling cascades that you're responsible for high detail of, right? So you have the weekend to work on it. And I made the other 2 sections pretty gosh darn short. Okay? Good. Any questions? Yes, Ross gets the funny joke. Oh, I didn't tell y'all a joke today. I have one. Okay, so what are Lego people's favorite dance? Square dancing. Oh wait, I have another Lego one, because my son's so into Legos, I had to look up Lego jokes. What is the most common surgery in the Lego hospital? Plastic surgery.
I got one.
All right, wait, we got one in the classroom.
I was at the bank earlier, and there was an old lady next to me. She said, "Can you check my balance?" So I pushed her over.
All right. Next class, yes. So the clicker questions that I'm going to go post this instant, as soon as I get back to my office, does have signaling cascade questions in there. They're all multiple choice, and you can work on them over the weekend. It can help you kind of solidify those pathways in your mind. I'm going to go post them like as soon as I get to my office. I'm so sorry it wasn't up before class. Okay. Yeah. So that the test can be moved to next. No, I'm not moving the test. We're having it on Wednesday.
I have a question about mixed inhibition.
Okay.
So whenever you have the 2 different,
Y'all have a good day too. If y'all have any other questions, yeah, just let me know. Put it on the chat, and because we have some other questions in the classroom too. So y'all have anything before I let y'all go on Zoom so I can end the Zoom? Yeah. Hannah? Let's see. Okay, I think I have the volume up. Maybe.
Okay, I'm sorry. For the video for 8.2 that we had to watch,
Yep.
like within like 21 minutes and 30 seconds, you said I have to find my paper first. If I can ask the question later, I have to
You
find.
can email me.
No, like I have my paper with me. I just got to read through it first.
She has a question, but she's finding her paper, reading her paper.
Okay. Okay, we're gonna work on this, but can I end the Zoom? Are y'all okay if I end the Zoom? All right, I'm gonna go ahead and end the Zoom. If y'all have any questions, send me an email.
I'm sorry. I do have my question now.
No, I'm not ending the Zoom. I'm ending the recording. That's
Oh,
what-
okay. Okay.
Just so that it doesn't go on forever.
Okay.
Okay.